So welcome everyone to our What's Happening in PWS Research Spring Edition webinar. We're excited to have you joining us today. I'm Susan Hedstrom, the Executive Director for the Foundation for Prader-Willi Research, and I'll be today's moderator. As always, we would love to hear from you. If you have any questions, please feel free to share them with us through the Q&A panel, and we'll answer as many of your questions as we can at the end of today's presentation. And with that, I'd like to hand you all over to our Director of Research Programs, Dr. Theresa Strong, who will be updating us on what's happening in PWS research.
Thank you, Susan. Great to be here today during PWS Awareness Month. So let me get some slides up and we can jump right in. So excited to join you today and tell you a little bit about what's going on in PWS research and also talk about the new grants that FPWR is going to be funding in spring of 2024. So we'll, as I said, do an update on the research activities, review the new grants, and hopefully have enough time to answer any of your questions or comments that you have. And just as a reminder, the mission of FPWR, I always like to start with that, is to eliminate the challenges of PWS and Shaft-Yang syndrome through the advancement of research and therapeutic development. And we do that by supporting research across the research spectrum. So everything from research and discovery, understanding, what's different in a PWS cell, to preclinical development, where we test new therapies in mouse and cell models, and then supporting clinical development or testing of new drugs and devices in humans, and post-approval, and that is taking approved drugs and making sure that they're used in the best way to support care of all individuals with PWS across the ages. And our goal is to accelerate every step of this process so that we get to that final healthy and happy individual with PWS. And we do that in a lot of ways. Today, we'll really focus on the FPWR grant program, which supports research across the entire spectrum. But I'll also highlight a little bit of the work of some of our research tools, including the PWS Clinical Investigation Collaborative, or the PWS CLIC, which is a network of PWS clinicians in North America, and the Global PWS Registry. So I just wanted to highlight three recent publications that really represent some of the work that we're doing here at FPWR. So that includes one from Dr. Sani Roy, one from an FPWR collaboration with the scientists at the University of Alabama at Birmingham, and one that represents a collaboration of FPWR and an industry partner. So the first one is from Dr. Sani Roy and colleagues at Cook Children's Hospital. And this is, again, highlighting the PWS CLIC. So the CLIC is a clinical investigation collaborative, so it's different clinical sites across North America, all of whom take care of individuals with PWS. And FPWR is providing a forum in which those physicians and healthcare professionals can get together and learn from each other and ask questions about optimizing care for all individuals with PWS. So it's been great to see this group has been together probably 2.5 years, and it's a very active group. There's a lot of different projects going on, but one of those came to fruition, and that was a question that Sani Roy and Jessica Dewis and others had about the use of feeding tubes in babies with PWS. So they wanted to understand, they were seeing some things in their practice, they wanted to understand in the broader picture what type of feeding tubes are being used and what are the complications that occur with those feeding tubes. So they got together with clinicians in the CLIC, developed a survey to try to get some answers to that, and then we collaborated with them and put that survey into the Global PWS Registry. And then all of you, about 395 of you, thank you very much, all who participated, filled out that feeding tube survey to give the physician some information about your experience with feeding tubes and any complications that you had. And this allowed them, because it was such a big data set, to really get a handle on the complications that were occurring. What they found, among other things, is that most babies needed feeding tubes, which was not unexpected. About 70% of them had NG or nasogastric tubes only, whereas 30% had G tubes either in combination with NG tubes or just alone. And what they found, and this was pretty unexpected, was that the severe complication rate from the G tubes was much higher than from NG tubes. About 25 times the severe complication rate. So, and that's not the same in all populations. I think it was somewhat unexpected. So it suggests that while G tubes may be appropriate for some babies with PWS, NG tubes probably are the first thing to look at for clinicians. So the nice thing about this is it has kind of gone full circle from a clinical question to collaborating with other clinicians and the patient community to get an answer. And at our last CLIC meeting, virtual meeting we had a couple of weeks ago, Dr. Roy presented this data and answered questions from the other healthcare professionals around, the country and in Canada about what changes they're making at Cook Children's Hospital to reflect the findings of this study. So this is going into, the publication is out, so any physician, who's taking care of a baby with PWS can see the publication, but also the guidelines and the changes that to NICU protocols are being discussed among the clinicians so that there can be continuity of care across North America, which is our goal, one of the goals of the CLIC. The second paper I want to mention is, as I said, a collaboration between FPWR and scientists at UAB. And you may have heard about this. I've talked about it before, looking at the whole genome. So we're doing a pilot study of about 50 individuals doing whole genome sequence analysis. And the goal is to really answer the question, how do DNA variants, both within and outside the PWS region, impact the severity and the scope of symptoms that we see. So we know some individuals with PWS have had scoliosis and some don't, and some have more mental health problems and some have less. So what is the contribution of DNA variants to those questions? So that's an ongoing project, but one of the aspects of it is returning results responsibly to participants. So as part of that analysis, we got information about pharmacogenomics, so how individuals metabolize different drugs. And for individuals or participants who were interested, we returned that information to them about their DNA variants. And then a genetic counseling student at UAB did a survey of people who received that information to understand, how they were planning to use the information, what they thought the challenges would be in sharing that with their physicians. And what she found is that most families were very eager to receive this information and learn from it. They were a little bit more unsure about if their physician was going to be comfortable and able to incorporate it into their clinical care. So keep an eye on this space. This is just a first step. Since then, we've done some additional surveys of this population to see how those families are sharing that and, whether their experience is good or not. And I think that will inform, any individualized pharmacogenomic studies moving forward. The third publication I wanted to mention is one that is just out about a week ago or so. We're going to write a blog and have it available on the website. But this is a study looking at a natural history study or Path for PWS study compared to a population of PWS participants that are receiving DCCR or diazoxide. So to take a step back, most of you are probably familiar with DCCR. This is a drug that has been evaluated in PWS and is on the cusp of being submitted for a new drug application for the FDA with the hope that it will be a newly approved drug for PWS, within the next year or so. So to think about the long term, this sort of PWS and DCCR started their relationship about a decade ago. So FPWR funded a study that Virginia Kimonis at UCI did in about 2014. And that was looking at DCCR in, it was a small study in individuals with PWS. It was a small study, but it showed some benefit, and it really prompted some interest from industry, additional industry partners. That led to Soleno Therapeutics expanding the study and moving it to a phase 3 study, the Destiny PWS. So Soleno Therapeutics did a phase 3 double-blind placebo-controlled trial, which is what the FDA likes, you know, a well-controlled trial. And that trial had positive results. However, life got in the way, and the pandemic occurred, and the results, which were strongly positive before the pandemic started, got muddied when the pandemic started. As you'll remember, early in the pandemic, it was, you know, it was a lot of disruption to people's routine, which could impact behaviors. So the FDA was really likes to see, you know, that drugs are very significant on their primary endpoints. So they were not ready to get a new drug application from Soleno at that point, but Soleno persevered, and they did an open label extension and gathered more data about the safety and efficacy of DCCR in individuals with PWS, which is very important. But you know, again, the FDA wants at least one well-controlled trial that has positive results for rare disease. They generally want 2 well-controlled trials for other disorders, but will accept 1 well-controlled trial with supporting information for rare diseases, and PWS is a rare disease. So what kind of supporting information could be used in the new drug application? Well, it happened that FPWR was funding a natural history study, and the FDA has encouraged the use of natural history studies to support medical product development in recent years, particularly. So the Path for PWS study is one that many of you are probably involved in, and I cannot tell you how much we appreciate your dedication to that study. It was a 4-year natural history study of individuals with PWS age 5 and up. And the goal was to look to document serious medical events, to understand what is sending our kids and adults with PWS to the hospital or the emergency room over a 4-year period, and also look at changes in behavior. We had tremendous response from the community with more than 600 participants enrolled. You all have been fantastic about entering data into the global PWS registry as part of the Path for PWS study. And it's really paid off with having a rich data set that now can be used to support drug development for DCCR and potentially for other drugs as well. Importantly, the evaluations that were used in the Path for PWS study are also the same that are being used in clinical trials of drugs for PWS. So this paper just came out, as I said, about a week ago, comparing the Path for PWS participants who are not receiving any experimental drugs to those in the DCCR open label extension who are receiving DCCR. And what we see over the long term, both half a year and out to a year, is that the individuals who are receiving DCCR, which is all the green bars, do much better as far as reductions in hyperphagia and improvements in behavior compared to Path for PWS participants who are not receiving DCCR or any other experimental drug. So this analysis was done by a third party. So we sent the de-identified data, and Soleno sent their de-identified data, and then a third party did the analysis. So this is, you know, an example of supportive data for potentially supportive data for a new drug application. So in aggregate, now DCCR has, you know, a small pilot study. They have their phase 3 study. They have data comparing the participants to natural history data. And then importantly, they also, after talking with the FDA, decided to do a randomized withdrawal study. That data was presented last October at the FPWR meeting. There's a link if you want to, you know, see Jennifer Miller's presentation on that. And that data was also very strong and well controlled, and showing that those treated with DCCR do better in many aspects of both hyperphagia and behaviors. So Soleno has received breakthrough designation from the FDA, which is very encouraging, which means that the FDA will accelerate the review once Soleno submits a new drug application. And if you would like to further support this, the PWS Advocacy Coalition, which is a coalition of groups and individuals who are interested in supporting PWS research and well-being, have a survey that you can, and a petition to the FDA encouraging them to review this data. So Soleno has said that it will be submitting their new drug application to the FDA by mid-year. So we're really excited about that. And I think the community is definitely looking forward to that review and hopefully a positive outcome with the FDA. So that's fantastic. I would just say, and you know, we can all pat ourselves on the back. I think, you know, everybody in the community in one way or another has contributed to this effort. And that's great, but I would argue that our work is not done. And rather than sort of sitting back and basking in the glow of that, we should really double down and get as many tools in the toolbox to help individuals with PWS that we can. So I have, you know, intimately known PWS now for about 30 years, because my son is now 30, which is incredible to me, but he is. And I have known many of you and talked to many of you over the years, and what I see is that PWS changes over time, that the severity and what is most bothersome or most difficult varies between families. So I think, you know, what we need as a community is as many interventions and treatments as we can find for PWS. So I would encourage you to stay up to date and consider the additional clinical trials that are ongoing in the field. All of this information is on our website, and there's many different studies that are either ongoing or going to be starting pretty soon that may be, you know, ultimately impact our ability to give our kids with and adults with PWS really the life that they deserve. So, you know, here's some of the ones that are either recruiting now or soon to be recruiting. And I think importantly, these different studies really address different aspects of PWS. So we know daytime sleepiness is a real problem for many kids with PWS. It impedes learning. What we saw in the registry study that we did with Dr. Dewis is that those kids who have the most sleep disturbances also have the most behavior problems. So addressing that is probably going to be very important. So maybe you want to look at the Harmony Biosciences pitolisant study for that. If you can't enroll in a medication trial or you can't get to a clinical site, maybe you'd like to look at the Maimonides study, which is entirely remote and looking at a bright light therapy to reset circadian rhythms. If your individual with PWS is struggling with hyperphagia and also with some of those anxious repetitive behaviors, maybe the drug carbetocin that Acadia is evaluating is for you. If your child is younger and you're interested in improving sort of overall development, you should maybe be looking at the Neuron study. If the individual with PWS that you love has a lot of temper outbursts, which makes them a little bit more difficult to love, you might want to learn about the VNS for PWS trial, which is a trial that FPWR is doing. And if your individual, you know, really has problems with hyperphagia, look for Aardvark Therapeutics will have a new study coming along soon that may address that. So there are all kinds of studies going on to address any of the problems that we see in PWS. And I would really encourage you to keep an eye on these clinical trials. New sites are popping up all the time. So, you know, maybe it'll be easier to get to one of them for you. And let's get, you know, as many treatments for PWS moving forward as we can. Okay, so I will get off my soapbox about clinical trials. I guess it's just, you know, I have the perspective, somewhat historical perspective now. When my son was born and when he was young, there really was nothing on the horizon for treating many of the most difficult symptoms of PWS. And now we have so many things, and it's really encouraging. But we, as Elizabeth Roof says, we have to get the butts into the seats to get the clinical trials done and get these treatments available to everybody with PWS. Okay, so we will turn to the FPWR grant program now, which is a very important part of our research programs because it supports all kinds of research that ultimately improves our ability to treat PWS. So we have, over the years, FPWR is now 20 years old, and we've invested more than 22 million into these various programs. And this has resulted in quite a few publications and advances in our understanding of PWS, new treatments. As we said, you know, the first clinical study of DCCR was supported through this grant program. It has brought new investigators into the field, which is critical to keep expanding our understanding of PWS. In spring of 2024, we're going to fund 9 new projects and invest that's about $1 million of investment into the research enterprise. And we've split this up into genetics and cell biology, which is the more basic science, understanding how loss of the PWS region genes leads to those characteristics that we see in PWS. The identification of new targets. So, you know, with the understanding of what changes in PWS, can we find treatments that will bring things closer to a typical cell, for example? And will that improve, you know, aspects of PWS in a person with PWS? And then advancing those therapies to clinical trials, either, you know, directly looking at them in clinical trials or doing work that supports clinical trials. So this is a list of the studies that we'll be supporting in spring of 2024, and I'll go through them one by one very briefly. So as I said, the genetics and cell biology is really understanding how loss of the PWS region genes, that's all the blue genes that are shown here, leads to PWS. And we do that not only because we just want to know, but because understanding that makes us smarter about trying to develop therapeutics. We don't want to be wandering around in the dark. We want to have an understanding of what these genes normally do, what happens when they're lost, and how we can kind of recover that function. So there are 4 projects that fall into this category. The first 2 of which are looking at these non-coding or SNO RNAs that are critical to PWS. So these are unusual kinds of genes in the genome, and so they're difficult to study, and so it's been a bit of a challenge, but these 2 investigators are really experts in looking at SNO RNAs. So David Tollervey has spent his life looking at SNO RNA function, and there are probably about 200 SNO RNA clusters across the genome, and but now he is turning his attention to the SNOs that are important to PWS, so SNORD 115 and SNORD 116. And he has, this is a second year of funding, so he has, in previous work that FPWR funded, showed that as neurons mature, so he has a model system where he grows neurons in a dish and makes them mature or differentiate. As they mature, the levels of SNORD 115 and 116 goes way up, indicating that, you know, maybe those genes are important in the maturation of the neurons. When he deletes those SNO RNAs, as occurs in PWS, what he sees is a change in the maturation timing and profile. And so that is very important. You can understand how a brain needs to mature in a certain way and with a certain timing in order for everything to sort of come out in a typical way. So the goal of his study is to use specialized techniques to understand the role of these SNO RNAs as neurons mature and identify the RNAs and proteins that these SNO RNAs are interacting with during that maturation process. So we're excited about this study because, as I said, Dr. Tollervey has spent his career really understanding snow RNA biology. He has recently applied his knowledge to the PWS region and has, already made some interesting insights and new advancements in our understanding. And understanding the normal role of these snow RNAs is very important for it. As I said, we have to understand how these things normally function so that we can figure out more efficiently how to correct for the loss of the snow RNAs. So the long-term goal of this study is to understand the normal role of SNORD genes in maturing neurons, and also find approaches to activate the PWS genes on the maternally inherited chromosome 15. The second project in this category is also focused on the snow RNAs. This is Dr. Gordon Carmichael at University of Connecticut, who has also spent his career looking at RNA structure and RNA biology. This is also his second year of funding. So Dr. Carmichael has used FPWR funding previously to look at cells with or without these SNORD 116s and ask what is different when you lose the SNORD 116. And from that work, he's generated a list of genes that change upon loss of the SNORD 116. And here he's going to investigate that more deeply. So we're excited about this work because he's a very experienced snow RNA researcher. There's an important question to be answered here, and he's had a very productive first year. And the long-term goal of this study will be to shed new light on the molecular mechanisms underlying the PWS, what happens when the PWS genes are lost, and identify novel therapeutics. The third project in this category is a new project from Dr. Gina Yostin, and it will look at spatial molecular imaging of the human PWS hypothalamus. So we know that many of the symptoms of PWS may originate in the hypothalamus, but if you look at a hypothalamus under a microscope from PWS and from a typical individual, they really don't look that different. However, we know that the peptide processing, so the process of making hormones and neuropeptides like oxytocin, is not quite right in the PWS hypothalamus. So Dr. Yostin is going to apply a new technology, spatial molecular imaging, I love the name, to examine the human hypothalamus in PWS compared to typical individuals at a very fine level, looking at the proteins that are in each cell, how that's spatially arranged, and what are the differences. Importantly, her team will also develop a data sharing platform so that information can be shared with all researchers who are interested in looking at this in the hypothalamus of typical individuals and those with PWS. So we're excited about this. We've funded Dr. Yostin before for an entirely different project that she's been doing great work on. Here, she's going to be looking at an important gap in our knowledge about sort of at the molecular level or at the cellular level within the hypothalamus. What are the differences in proteins? And, she's already thinking ahead to how she's going to share that so it can be leveraged by everybody in the PWS research community. So the long-term goal is to provide the scientific community with a spatial proteomic and transcriptomic map of the PWS hypothalamus, and do that in situ. So right within the hypothalamus where the proteins are sitting spatially. The final project in our sort of cell and molecular category is defining isoform diversity conserved in the blood and brain and related to severity in PWS. So Dr. Godler has been looking at brain samples from individuals with PWS and comparing that to typical individuals and asking, again, what are the differences? But he is also looking at that in the blood of individuals with PWS and asking if you can use the blood to understand what's going on in the brain. And that's obviously important because we don't want to go sampling a person's brain. That's not, it's not nice, it's not feasible. But if we could look at their blood, we might be able to identify those people, for example, who are at highest risk for, some of the issues that we see in PWS. So the study goal here is to analyze brain samples, understand one of the proteins that he has seen be different between PWS and normal, these ribosomes, how they influence the differences between PWS and typical individuals. So we're excited about this project because Dr. Godler is an experienced PWS researcher. He has strong preliminary data. He's using cutting edge technologies. And, it would be really nice to have some markers of severity that we could look at in the blood of individuals with PWS to identify who's at risk for, what aspects of the disorder. So the long-term goal is to define the genetic pathways that are dysregulated in blood and brain from individuals with PWS, and use that information ultimately down the line to inform, drugs that may correct these differences in PWS. Okay, so now we're moving on to the target or the preclinical studies using mouse models, usually of PWS, to understand neural circuitry, for example, and think about how we can apply therapeutics. So we have two projects in this category, and the first is from Rachel Ross. This is also a second year of funding. So Dr. Ross is really interested in the connection between cognitive ability and food drive and food behavior. So how do those interconnect in someone with PWS? So she is using a mouse model of PWS, the MAGEL2 mouse, and looking at a particular neural circuit, these prefrontal cortex glutamatergic neurons. And I'm not a neurobiologist, so I may have said that wrong, but she's looking at a particular subset of neurons that are important both in cognition and in food awareness and food impulsivity. And she wants to test the hypothesis that this molecularly defined subset of neurons in the prefrontal cortex are involved in both cognitive function, the cognitive function related to food intake, and food-related impulsivity. And that perhaps if you specifically stimulate this subclass of neurons, you can modulate both cognition and food behaviors. So we're excited about this study because, again, we had initially funded Dr. Ross, and she's made excellent progress and some new insights into that connection between food and cognition in PWS. She is an enthusiastic researcher. She came to the conference, gave a great presentation in Denver last year. And she's applying some innovative approaches to the PWS mouse models. Her long-term goal is to find druggable targets to develop novel therapeutic approaches that might impact both hyperphagia and cognition in PWS. The other preclinical study is from Dr. Azevedo, who is in South Carolina, and she has done some previous work that shows that a particular set of neurons in the lateral septum connects to the hypothalamus, which is that brain region that is important for regulating hunger, and that those neurons strongly express the PWS region genes, including MAGEL2, which we know is an important gene in the PWS region. So she wants to investigate whether these neurons behave differently in PWS mice compared to typical, or what we call wild type mice, and potentially identify a new circuit that can be targeted for therapeutic development. So we're excited about this investigator. She's a promising early stage career researcher, new to the PWS field. And that's really one of our goals, is to bring promising new investigators into the field. She's looking at a brain circuit that really hasn't been very well explored in PWS, but it makes sense that this would be an important circuit in PWS. And there's a high potential for translatability if we can define this as an important brain circuit for PWS. ultimately, again, she wants to find a druggable way to influence this brain circuit and show its importance in PWS. The final category that we'll be looking at is studies that support therapeutic development or clinical trials, and there are 3 in this category. The first in this category is from Dr. Richard Lee at Johns Hopkins, and he is looking at in vivo implementation of hypothalamus-specific exosomes to reverse the impact of SNORD116 deletion. So what he's doing, he's looking at exosomes, which are these tiny vesicles that come off of cells. And exosomes lately have been considered as a really potentially really interesting gene delivery vehicle. So a lot of times for gene therapy, we'll use viruses, and they have some advantages, but they're viruses, so they have some downside as well, like an immune response. We're very good at making immune responses to viruses. Exosomes come right from cells, and they can package genetic material, DNA or RNA, and they can, depending on what's in the exosome, they can deliver to different parts of the body. And so Dr. Lee is looking at exosomes that are specifically targeting to the hypothalamus as a gene delivery mechanism for PWS. So he was funded previously to look at this, and he has done a lot of nice work in that area. And here he's going to be using their engineered exosomes to test whether delivery of PWS genes, specifically the SNORD116s, can reverse some of the features seen in PWS mice. So we're excited about this project because it's an exciting and innovative new approach. The gene delivery approaches to the hypothalamus are pretty limited, so this may give us a new option. And he has been productive in previous FPWR funding. And the long-term goal of this study is the implementation of exosome technology for gene delivery in PWS mice, and this might lay the foundation for using that same technology in human gene delivery approaches. The next study is really one that is looking at instruments or measures of some of the behavior and psychiatric problems that we see in PWS, and considering whether some standard questionnaires that have been used in other settings are applicable to PWS. So we know that people with PWS have a range of behavioral challenges, and that can vary by genetic subtype or maybe other things like, gender or age. But we still don't have a very clear risk. We can't tell sort of who is at highest risk for developing specific problematic behaviors and mental illness. And we'd like to be able to predict that so that we can intervene early and prevent there from being a problem. So this study is going to examine repetitive behaviors and obsessive compulsive behaviors using validated instruments. So questionnaires that have been used widely in in typical populations and other developmental disorders, but which have not been explored in PWS. And this is going to use the global PWS registry. So once we get these in there, we will ask you to, fill them out for your children. And the goal here is to see if they, again, if these are useful instruments in the PWS population, and if they can, predict who is going to have the biggest challenges. So we're excited about this project because it raises the possibility of having a more individualized approach to understanding who is at high risk for behavioral problems or mental illness. It's using a validated instrument so we can compare to other populations, and it's an experienced researcher. It's a researcher who is new to PWS but has a lot of experience in this field. So the ultimate goal of this study is to understand key variables that predict behavior trajectories in people with PWS so that individualized interventions can be developed. And I believe this is the final one, is looking at cross-cultural adaptation and validation of a food attentional bias task as a potential biomarker for PWS. So we know that measuring hyperphagia in PWS is very challenging. What we rely on now, we being the entire field of PWS, in clinical trials is a hyperphagia questionnaire for clinical trials, which has strengths, but is also, has its limitations. So it would be very nice to have an unbiased way of measuring somebody's attention to food or focus on food. And Dr. Gallagher, who we've previously funded, has been looking at an eye tracking method to look at what people look at on a display and use that as a measure of how much focus they have on food. She's had good results in her initial studies and now wants to expand that to new populations. So she was originally, she's from Ireland initially and did her initial studies in the Irish population, but is now in Canada and will be doing additional studies with collaborators in the US and in Canada. So we're excited about this study because she was very productive in the first year. The studies that she has done look promising for their potential, perhaps to be used in the clinical trial setting, but you have to show that it can work in more than one population in order to be widely used in PWS clinical trials. So this is an important step towards that eventual goal of perhaps using this as an alternative method, a more unbiased method of understanding hyperphagia and food focus in PWS. So that is all of the projects that we'll be funding in this round of funding. As you may recall, we do 2 rounds of funding per year, so we'll have another round in the fall that we'll tell you about. But there's also a lot of upcoming research activities. The Global PWS Registry is going to be moving to a new and improved platform, so it's going to get a bit of a facelift. We'll be adding some, as I mentioned, new surveys based on some of our new studies. We look forward to engaging you, continued engagement through the Global PWS Registry. I hope you're seeing how important the data that you put into that registry is, because when we can get 400 or 500 or 600 people to put their data in there, we really can gain an understanding of the challenges that we see in PWS. FPWR is, we are rolling with our vagus nerve stimulation clinical trial. So if your loved one with PWS is between the ages of 10 and 40, lives, it has a lot of temper outbursts and, might benefit from reducing those outbursts, please contact us and we can tell you more about the VNS for PWS study. On the clinical trial front, as I mentioned, Soleno will be submitting its new drug application to the FDA, and we're excited and have our fingers crossed and are knocking on wood and whatever else superstitious behavior you'd like. Hopefully that drug will move forward for the treatment of PWS. And we ask you to save the date. We'd love to see you in Atlanta in September. We're excited to get together with the PWS community in Atlanta this year, next June in Arizona. So lots of great meetings coming up. And I will end there and thank you for your attention and see if there are any questions.
Well, there's always questions. Theresa, thank you so much for making the research understandable for people like myself who perhaps don't have the scientific background. One of the projects that you were mentioning I thought was quite interesting, this exome delivery system. You mentioned the cells package up genes in these exosomes and they send it out to the rest of the body. Why are they choosing SNORD116 as a gene to deliver versus MAGEL2 or another gene?
Yeah, I mean, the nice thing about the exosomes is kind of you can put whatever you want in there. I think the SNORD116, we know that loss of the SNORD116 occurs in every person with PWS. So they're kind of they're a really important target gene. So I think, you can see that project potentially in the future expanding to look at, if Magel-2 only delivery, can recover some function, and comparing that to SNORD116. I think this is just to get them started, and they can track the SNORD116 getting into the hypothalamus. So it's the first step in what could be an expanded program. Like I said, the nice thing about the exosomes is you can put lots of genes in there. You can put all the genes in there, all the genes in the region in there. And that is not the case with many of the viruses. A lot of the viruses have packaging capacity issues that you have to be more careful. So it does have the potential to give you more flexibility in that.
Well, that's really exciting. I know gene therapy has been something that we've spent a lot of time focusing on, but as you mentioned, those limitations are a challenge.
Yeah.
So more tool in our toolkit. You talked about a researcher who's going to be adding a few new surveys to the registry. When will we be able to start completing those surveys, and are they targeted for a certain age group?
Yes, that's a great question. So I think what we're going to do is we're in the middle of the registry migration. And so what we hope to do is have that moved over during the summer. And so you should look for these new surveys, hopefully in the fall. And I think it does go down to a certain age. Off the top of my head, I don't recall. I mean, I know it goes down to younger children, but I don't recall the exact age. So I'll have to get back to you on that one.
Great. We have a question regarding timelines of when drugs become available. And this is a question that always comes up. So perhaps you and I could spitball a little bit in regards to specifically carbetocin. Carbetocin, they just opened up a new clinical trial. They're still opening additional sites for these trials. Certainly, the faster they can enroll the study, the faster they'll have answers. But a phase 3 trial is, a year, 18 months is typically how long it's taking for them to complete. Is that about right, you think?
Yeah, probably because the treatment period is usually, and I don't remember exactly for the carbetocin trial, it's probably 3 or 4 months maybe. So the treatment period is 4 months, and then you have the open label extension. And it takes a while to get all of the clinical sites up and going, and just scheduling can be a problem because, these physicians have a lot going on. And so being able to fit everybody in. So what we don't want to happen is that the enrollment is what's holding us back. There's plenty of things that are going to hold us, slow the whole process down. I mean, as I was putting these slides together and I was looking back, I looked back, what was the year that we started we funded the first DCCR pilot study? It was 10 years ago. A lot of things happened, COVID and everything, and we couldn't have predicted that. So these things do take a while, but we don't want recruitment to be the thing that's slowing us down. So anything we can do to, have people, at least consider, like, none of us want you, like, that is a personal decision, whether you have the capacity and whether you're comfortable enrolling a clinical trial. But what we ask you to do is learn about them, see if they're near you, consider it.
There's so much out of our control when it comes to clinical trials. But the one thing that we as a community do have control over is whether or not we enroll our children into these studies. So the faster we get the studies filled, the faster it can get reviewed by the FDA. Thank you so much to the anonymous person who submitted that question. So Theresa, back to gene therapy replacement and research. This is the potential to be transformative for people with PWS. When we start talking about gene therapies, this is perhaps, we don't like to say the C word, curative, but it's as close to a cure as we're ever going to get. What's the timeline for this miracle? Is this, short term? Is it long term?
Yeah, I mean, it's probably honestly still long term. So there are a lot of different approaches that are being evaluated. Some of them are doing like activation of the maternal allele that every person with PWS has. So every person with PWS has a chromosome 15 that they got from their mom. All of the genes are sitting there and they just need to be turned on. So there are people who are trying, drugs to do that, and gene therapy approaches to do that, and CRISPR approaches to do that. Now, the CRISPR is probably the furthest away, but CRISPR trials have started, so there'll be experience that is gained in more devastating disorders. So disorders where, people that are lethal, right? So those will be the first disorders that, use some of the newer technologies. And that experience in the field of gene therapy will be important for us to learn from, because, we're safety wise, we probably want to have a pretty high threshold. We want any gene therapy that we use to be, quite safe before we use it. So I'm not sure if I'm answering your question about timeline, except to say that there's a lot of different approaches. They're all being evaluated. The most safe ones will be the first ones that go into clinical studies. And those are still, at least 3-4 years away, I would think.
Our next question is just, it seems that there's more clinical trials now than there were perhaps a few years ago. Is that observation correct? Are there really more clinical trials today?
Yeah, I mean, I think the number of clinical trials has really been increasing over the past 7 or 8 years. I think that there's a lot of things that play into that. I think there's an increased understanding of, the ways, why people overeat and things like that. I think the fact that our community now has, good natural history data, that companies know that we're engaged, that PWS has a very robust diagnostic tool. So, you can say yes, no, whether a person has PWS. All of those things are important. And then the engagement that we've had, for example, with the FDA, so that the FDA understands the unmet need in this population. I think all of those things have played into, why there is so much more activity in the PWS field now as compared to a decade ago.
We have time for just one more question, and I think this one's interesting, perhaps top of mind of a lot of people, and that's in regards to Ozempic. Ozempic is, a hot word these days. So many people are trying it. Where do you think Ozempic fits in when it comes to people with Prader-Willi syndrome?
Yeah, that's a great question. I think I'm using the royal we, the clinicians are going to gain some experience with that. So Ozempic is already being used in, older individuals with PWS and diabetes. So there'll be some knowledge, and the CLIC members have already been talking about that. How can they pool their collective experience to, really understand who is best suited for being treated with Ozempic? I think that Ozempic works really well in some people and less well in other people when it comes to weight loss. We know that many of the earlier GLP-1 inhibitors like Byetta and things really didn't result in a lot of weight loss. So it remains to be seen if Ozempic and Lilly's drug tirzepatide, which adds a different, it's sort of like a twofer, how useful they're going to be in PWS. I think obviously it's really important to understand that. I think they're only going to be useful in those people who are overweight. So I, at least, and I'm not a clinician, but I can't see them being used in, normal weight individuals, at least in the short term. And we have to watch things like we know individuals with PWS have a slow GI tract, and these drugs may slow it down a little bit more, and that may not be a good thing. However, if there were really good weight loss, you would look at the risks and benefits, and for many individuals, it might be a way to go. So I would say that the clinicians are interested in understanding how these new drugs can be used in PWS. And so I think we'll be seeing some studies, either, full-on studies comparing Ozempic to, something else, or just gathering data of people that are using these drugs to understand. So I think it has the potential to be another tool in the toolbox, but like I said, it's not likely to be perfect for everybody. So we still need to be looking for additional treatments to help our kids and adults.
That does sound key, Theresa. You know, even in looking at clinical trials, we're seeing that some individuals are responding more than others.
Yeah.
Clearly, there's no single drug that is best for everybody. So having as many options as we can is so incredibly important for the community as a whole. I'd like to thank you, Theresa, as well as everyone else who joined us today for this webinar. May is PWS Awareness Month, so please continue to share facts about PWS throughout the month. Let's help spread some awareness. There's the petition that you can sign should you like to encourage the FDA to review the DCCR application. And of course, if you want more information, you're welcome to visit our website. We've got lots of information over there.
Susan, hey, sorry, can I say one more? I do see one more question, which is how important is fundraising in our research efforts? And I will just say it is absolutely critical. So, you know, the way that we can attract new researchers into the PWS field, the way that we can support the clinical network and run the registry is all because of the fundraising efforts of our community. So, you know, I cannot thank all of you who do fundraising enough, because it allows us to do the things that hopefully move these treatments forward.
You know, couldn't say it better myself. Everything that was presented on today has been funded through fundraising efforts from our community. So thank you, all of you who have done so, and thank you for joining us today. This webinar has been recorded and will be made available shortly. Have a good one.